Hello, everyone. Thanks for coming in to this big, exciting webinar. We have a big group of panelists here. We'll definitely start going through our slides, and we will have a session for Q&A after we finish the presentation. So please just start submitting your questions using the Q&A functionality at the bottom of your screens. And with that, thanks for coming. And Dr. C, maybe you kick us off.
Yeah, thank you everybody for joining. My name is Sajjad Zalzala, Chief Medical Officer, co-founder of AgelessRx, and the clinical investigator on the Pearl trial. Today we're going to be presenting the results and having discussion about it. I think I'll be driving the slides. We'll be talking about introductions, background rationale, study design methodology, key findings, implications, conclusions, some acknowledgements, and then, of course, most importantly, audience question and answer. That's why hopefully you're all here to focus for some questions. Oliver, do you want to go ahead and give us like an round of intros?
Yeah, sure. Okay, so yeah, welcome everybody. So as you mentioned, welcome everybody to this webinar, which is on the Pearl trial, participatory evaluation of aging with rapamycin for longevity. So joining us are Dr. Matt Kaeberlein, who's the co-founder and CEO of Optispan Health. He's also the founder and co-director of the Dog Aging Project, and he is currently the CEO of the American Aging Association and served on the board of directors for the Federation of American Societies for Experimental Biology, FASEB, AGE, and GSA. And also welcome Dr. Do you go by Dr. Z or Dr. Zalzala?
Zalzala tends to be a little bit of a tongue twister, so Dr. Z will be fine.
Okay, so Dr. Z is the co-founder and chief medical officer of AgelessRx and serves on the board of the International College of Integrated Medicine and is chair 2 of their international conferences. So you've also been involved with multiple successful healthcare startups, including Pill Club, Jack Health, For Hims, and Modern Fertility. So welcome everybody to this panel discussion. Also joining us is Keith Comito from Lifespan.io, and I'm, of course, Oliver Medvedik from Lifespan.io as well. So welcome, everybody.
Yeah, and on the AgelessRx team, we also have Garish Harnath. He's actually currently overseas at ARDD, so Denmark, having a blast there. And we also have Stephanie Morgan. She's a recent addition to our team. She's leading our research efforts, our applied science efforts, and she's the one who kind of led the charge on the data analysis. So you might hear them chip in as well. All right, we got a lot to present here, so I guess let's go ahead and jump right into it. So we're kind of go over the background and rationale. I don't think I have to spend too much time on this, but for those who may be unfamiliar, but targeting biology aging is key to longevity. Aging is by far the biggest risk factor for any chronic disease or mortality. Basically, whatever most of what ails our society these days. And then the rate of biological aging determines both lifespan and health span, and it's thought to be modifiable through various interventions. Over the last couple decades, or we've been looking more and more at rapamycin, specifically low dose intermittent rapamycin, as a key gerotherapeutic to help potentially mitigate age-related decline, to extend at least health span and maybe lifespan. Rapamycin, actually, I'm gonna let Matt, because he's kind of the world expert on on rapamycin. I think it's only fair that he kind of talks about this.
Sure. All right. Nothing like throwing me on the deep end, unprepared. Yeah. So probably as many of you know, I have studied rapamycin for a long time. And part of the reason why I've studied rapamycin for such a long time is it really is I think still the gold standard for pharmacological intervention that can positively modulate the biology of aging. We being the broader research community, have seen that rapamycin treatment or genetic knockdown of mTOR in pretty much every model organism where it's been tested in the laboratory, in fact, in every model organism that I'm aware of where it's been tested in the laboratory, increases lifespan and seems to broadly delay the onset and progression of declines that go along with aging at the functional level, tissue, organ, and also at the molecular level. So I think there's a lot of evidence that rapamycin positively modulates the biology of aging. I think one of the big questions and the whole sort of point of the Pearl trial is, how effective is rapamycin going to be at positively modulating the biology of aging in the real world? Many of you know I've been involved with a clinical trial of rapamycin in pet dogs as part of the Dog Aging Project designed to assess that question. And obviously the Pearl trial is one of a few initiatives designed to try to assess that question in human beings. So there have been a few sort of small clinical trials so far, but at this point, I think the only strong evidence for functional improvements in humans are the clinical trials done by Joan Manick with a derivative of rapamycin called everolimus, where they were able to show that in healthy older people, 6 weeks of treatment with everolimus was sufficient to boost influenza vaccine response. And I think interestingly, there really had very little in the way of side effects when dosed in a once weekly manner, which I think also sort of helped guide the design of the Pearl trial on some of the other studies that are ongoing. Broader understanding of gender-specific effects and optimal longevity dosing for rapamycin is needed. Absolutely. I don't really have anything to add there except to reiterate that we really don't have much in the way of evidence for what is the best or most effective strategy for dosing rapamycin in humans to hopefully have positive impacts on health while avoiding some of the side effects that we know can occur in organ transplant patients who've been taking the drug at high doses on a daily fashion. So people have gravitated towards this once a week dosing schedule. We can come back to it, I think, in the discussion of whether or not that there's a lot of evidence to support that, I think is a little bit of a question at this point.
Absolutely. Well, thank you for that. So that kind of brings us to, kind of these open questions kind of brought us to the Pearl trial. So this trial was conceived about 5 years ago, right before the pandemic, and the reason for it was let's see, do I have it in here? Sorry, we changed a lot of the slides at the last minute, so I apologize. But the reason for it, to give you a little background, is there's a very strong interest with the longevity community to have rapamycin prescribed, but to Matt's point, not very many studies to kind of support the dosing. There were some concerns about safety, things like that. So we set out to kind of design a trial that it would at least help explore the dosing for rapamycin, look at some of the safety markers and see what we could find. So it's very much designed as a kind of a safety and somewhat efficacy trial, at least from some of the endpoints. And I'll talk about the endpoints in a minute. But so far, it's been, one of the largest and longest, as a previous slide showed. Most of the studies on humans have been kind of short and very narrow. This is one of the first crowdfunded human RCTs, and thanks to Lifespan for that. And that's why we're thrilled to have them host us today. And then we were kind of looking to evaluate a broad range of healthspan metrics in healthy aging individuals, free of major disease, and as Matt said, collect real world evidence, not some mice in a lab, but humans out in the wild, so to speak. And so this was only meant to be kind of like the first of hopefully longer and larger trials. Some of the questions I could talk about, safety of kind of the longevity dose for rapamycin. In this case, we kind of decided to look over it for a year to see what we'd find. We wanted to test different doses, and then also enroll both male and female patients to see if there's any sex differences, and then look at different physiological aspects, and then we were hoping to we kind of knew going into this that some people responded pretty well to rapamycin based on kind of anecdotal, and some people didn't seem to respond very well to it. So we were hoping to kind of understand that a little bit better, or at least the beginnings of understanding for that a little bit better. So the big question, does rapamycin show potential as a safe and effective health span enhancing intervention in normative aging adults? So that's a mouthful there. So the study design, it's changed, for those people who've been involved in the study from the beginning. We had a different study design, and I don't necessarily want to go into that. But we ended up, we wanted to enroll a larger number, but there was a variety of different reasons, and I'll talk about that in a minute. So ended up finishing at 115 individuals between the age of 50 and 85. And like I said, we wanted a study that could look at safety, see how well tolerated, and look at various different changes in biomarkers, and then kind of efficacy or health span metrics were almost secondary in this study. And we wanted to be a 12-month study for 48 weeks. And we initially looked at 5 milligrams and 10 milligrams. And that was based on discussions with various different experts in the field, doctors who prescribe rapamycin, researchers in the longevity field, things like that. That did change from the initial study design, but that's where we ended up landing. And I'll talk about the dosing in a minute. Like I said, a secondary objective was look at more of the efficacy. And in terms of the clinical endpoints, we had to pick a validated clinical endpoints. It couldn't have been like, a biological clock, for example, or an aging clock, because those aren't really validated endpoints. So we rocked our head and kind of tried to figure out what would be an appropriate clinical endpoint. And we came up with visceral adipose tissue based on a smaller scale rapamycin trial done by a group called Better Humans. And I don't think they ever published it, but one thing that they told us came out of that trial was they saw an improvement in visceral fat. That was like the one thing that they were most confident would be beneficial. We didn't have any statistics or anything to go based off of, so we just took the best guess we could based on the very limited data. We were also looking at adverse effects, metabolic, and safety biomarkers. And of course, we have a bunch of secondary endpoints. The DEXA scan, can get a lot of data. We looked at, various different methylation clocks and fetal weight clocks and things like that. And of course, we wanted to also look at quality of life surveys. So included the SF-36 and the WOMAC for joint health. We're not going to be able to cover all these biomarkers today. So today we're kind of going to be giving the highlights. Now, to start out with, I kind of want to set some expectations. So because the nature of the trial in terms of being decentralized, we drew from a very enthusiastic crowd. These patients had a very high baseline. I mean, it seems like across the board they were doing everything they could to stay as healthy as possible. And so we kind of came up with this health composite score. And you can see the majority of our patients scored very high on this this health composite score, as well as very high on the quality of life assessments at baseline. So kind of going into this, we kind of had a little bit of uphill battle because these participants were already very healthy to begin with. At baseline, we did blood work, DEXA scan, and then the surveys. We asked participants to do blood work again at 2 and 4 weeks, so we can kind of monitor them pretty closely while they started to ramp up. And then we did a 24-week and a 48-week testing as well. And I kind of already mentioned what we looked at. At the end, we ended up with this is a randomized double-blind trial, so we didn't know who was on what group and what dose. We didn't know, the participants didn't know. So in the end, we ended up with 40 people in the 5 milligram, 36 people in the 10, and 39 in the placebo. So numbers worked out pretty well. We enrolled a little bit more than this, but of course, just like with any trial, we had attrition, which is why I wanted a larger trial. But there's a bunch of things conspired against us to make this into a larger trial. We designed this as a decentralized trial before the pandemic. And so a lot of people were like, oh, you can't do a decentralized trial. I was like, well, why not? But then, of course, during the pandemic, a lot of organizations moved to decentralized trials. So, in many ways, we were kind of ahead of the game. If only we knew the pandemic was coming, probably would have waited it out. Anyway, so we didn't really have much to go based on in terms of like power and efficacy and things like that. So this was very much an exploratory trial. And it turned out that about a third of the way through the trial enrollment, there was an issue that came up with the bioavailability of the non-encapsulated rapamycin that we were using. So it turned out, even though we wanted to do a 5 and 10 milligram, it turned out it was probably closer to like 1.5 and 3.5 milligrams, based on another study we published a couple of weeks ago, actually. So if you want to know more about kind of the difference between the encapsulated and the non-encapsulated form, there's another webinar, another study I encourage you to explore. But that was yet another challenge that we kind of faced. And of course, we talked about kind of this healthy user bias. If somebody's at the top of their game, so to speak, it's always hard to see improvement. But I think we did achieve our goal in being able to kind of move on to larger, more vigorously designed clinical trial. So the data gave us the confidence to expand. So what are the key findings? First, I'll talk a little bit about safety. We didn't really see much in the way of any rapamycin specific adverse reactions. If you look at the kind of the adverse reactions, that there wasn't any statistical significance between the three groups, and you can see by this chart here, actually, for COVID, there were some, again, not statistically significant, but some clues that maybe rapamycin could be protective. And this is also was seen in a registry, like an observational registry trial that the University of Washington completed and we helped participate in. So again, neither study showed statistical significance, but you can see that there are some clues here that rapamycin could help against it. And side effects didn't seem to be rapamycin specific. Keep in mind that this was a trial run during a pandemic. So we had several waves of COVID come through during our trial. So it was really hard, because we were blinded. We couldn't tell until the end, until we unblinded, what was who was on what dose. We just had to keep track of all the side effects and just kind of monitor for any serious one. Luckily, no major serious ones were seen, or were seen in the rapamycin to kind of that were thought to be blamed on rapamycin. Interestingly, the most common rapamycin-specific side effect was gastrointestinal upset. That could be because of the lower bioavailability. That's just my guess. It could just be noise. It's hard to say, but otherwise, there didn't seem to be anything different about the various groups, which was kind of reassuring that these doses seem to be well tolerated. All right, what were the results? So we looked at DEXA scans, and again, I mean, because the study ended up smaller than we liked, there wasn't any large statistically significant differences, but there were trends towards the 10 milligram rapamycin group having a little bit more consistent benefits than the placebo group. And we'll talk about the lean tissue mass improvements seem to be the most robust out of the measurements. But again, I mean, I want to emphasize that it's hard to say because of the limitations of the design and the small groups that we enrolled. So here's some of the graphs that we show. So the placebo is in this darker black color here. So this is female versus male. And you can see that kind of as expected, with the 1 year passing, visceral adipose tissue would go up, and you'd see then lean tissue also go down with every year that passes, especially in this age group, because after you hit 50, that's typically when aging signs of aging start accelerating. So you can see that so there was less of a increase among the rapamycin users. Again, I know the bars overlap too much, but again, I think it's useful to say that, hey, there could be something here. Bone mineral density, a little bit noisier, not very useful in this context, probably because it takes too long to see changes in bone density. You don't expect to see much in the way of bone density in a year. For anybody who's ever like treated osteopenia or osteoporosis, usually you don't check, you don't check in a year. You usually check, 2 or 3 years, plus limitations of the body composition versus the diagnostic bone mineral density. So again, maybe interesting, maybe just noise. It's hard to say. This chart here shows that there's a lot of individuality, or some people had a big increase, and some people had big decreases. So it's really hard to say. Interesting overall, female, we had at least one female age group have pretty big declines in bone density, and I would suspect that this probably has something to do with that menopause transition, because we were kind of like, for women in that age group, especially 50 to 60, I mean, that's kind of when they're known to like lose a lot of bone density during that menopause transition. So it could be why we saw that here. Now, of note, we did follow up with a participant. There was one female participant who actually had a diagnostic bone mineral density before the study, and then when we finished the Pearl, and then she continued on rapamycin for a year after, and so she was 2 years on rapamycin, and she actually saw a 15% increase in her bone mineral density, and this was a diagnostic test, and she had tried other things before. That's something worth double clicking on, I think, at some point, and investigating a little bit further, and see if there's something there, or is this just like a one-off event from a patient. So in clinical practice, usually trends typically start with one patient and then move on. If you see it over and over and over again, then and then of course that typically launches observational studies, and that then you move on to more rigorous trials after that. In terms of lean tissue, this is kind of where we saw the most robust data, especially in female participants at the 10 milligram group. It was statistically significant. Unfortunately, we didn't enroll as many female patients as we'd like, for a variety of different reasons. And I wish we actually saw that with the observational trial as well, that registry trial. I think of the 333, I think only 70 were female. So we had similar ratios in terms of male to female. I think, well, at least it was heavily skewed male, and I'm not sure why. But for the female trial, all of them, all 6 of them, here's kind of a breakdown. You can see all 6 of them in the 10 milligram group had improvement in their lean muscle tissue. And then again, like I said, kind of among the placebo and the other groups, you can almost, if you kind of look at it, you can almost see that, there was more decline than improvement in the placebo groups. And the 5 milligram just kind of seemed to be, evenly spread out. Again, this highlights the reason why we need to follow up. And Stephanie, can you tell us a little bit more about this? I know you put together this graph. If you want to like tell us a little about why you put this chart together for us.
Yeah, happy to. So we found, like, as we were looking through the data, we saw that we had these differences in response. And while it wasn't something that we wanted to really harp on too much or make big claims about, we did want to understand what was different. We saw, if we looked into the data in a deeper way, that there were patients that did really well on rapamycin, and then there were also people who had not a lot of response. Nobody really seemed to have any detrimental effects that raised concern for any of us, but we wanted to know what was different. And so we just looked at, we made a graph in all the different conditions, who improved, who declined, and we wanted to ask what might be different between those groups.
Yeah. So that just kind of visualizes, and you can kind of see among the 10 milligram users, more of the participants improved than decline. Again, I mean, it might be stretching the data a little bit, but we're trying to make the best of what we can with kind of a limited data set, and hoping to inspire people to kind of run with it and expand it. What about what was this? We had some interesting data from our Thorn partner for the microbiome. Could you go over this slide as well?
Yeah, so similarly, like just trying to take the data that we had available and try and figure out, what are good directions for future studies and what are things that we might follow up on in subsequent research from the data that we had. And so if we, our study was not powered in the beginning to really find significant effects. It was really more to evaluate, at these doses, is it safe enough? But we took those people who improved or declined just to see, like, if you respond to rapamycin, can we identify any markers that might suggest there's some kind of loop in should we pay more attention to this in the future to help recommend if you will benefit, or something to dive into in more depth molecularly in the future. So we had some data from some partners about measures of gut health, your gut microbiome. And so if we looked at the people who did well on rapamycin and saw a lot of improvement overall, they tended to have an improvement in their gut dysbiosis, which is like their like healthy bacteria. So you want to see like the unhealthy bacteria rather. So you want to see it actually goes down. So down is better in this case. So the 10 milligram group, the people who improved had more like healthier scores in the gut dysbiosis. And then there also was a measure in the gut health panel for immune readiness. And we saw that that improved just a little bit for the 10 milligram group, but it went down a lot for the placebo group. And certainly, doing this during a global pandemic where there were also a lot of other people, you stayed home for a while, a lot of people got really sick afterwards. This is small, but perhaps interesting as a future direction. Don't want to make too much out of it. This was really just for us as curious scientists to say, like, what could we kind of tell people we noticed as something interesting in our data that they could go do more research on in the future. And these are just some general suggestions about things we thought were kind of interesting facets of the data. Importantly here, like the analyses that we ran there is no directional information. So we don't know if people responded better to rapamycin because they had this change that was ongoing through some other diet or lifestyle change, or whether rapamycin drove this. So we don't want to in any way imply that this is causative. It was just an interesting nuance that we wanted to highlight because we kept getting the question from people as we talked about this. Well, what next? Like, what do we go after in terms of something that's a little bit more granularly molecular? And so this is one of the things we wanted to just share with people who might be curious about where to go with future research directions.
Yeah, interestingly, we actually saw a drop in LDL. There's been a lot of concern about rapamycin causing an increase in lipids. And actually, we saw the opposite. We saw 20% drop, or at least I don't know what the exact magnitude is, but in this chart, it's reading at about 20%. But that was kind of reassuring to us that we're not disturbing lipid metabolism like a lot of people were worried about going into it. We do have a larger data set for our patients through AgelessRx that we're going to be digging through in the near future, which might give us more clues to various different biomarkers. Let's see. We also looked at quality of life. So this is the SF-36. It looks across 8 domains. And again, in this case, we started with really high baseline scores, so it wouldn't have been unreasonable to expect that we wouldn't see any change because of the high baseline. But female patients did seem to drive the improvement in quality of life, whereas males, it was kind of hit or miss for male participants. This is the WOMAC score. It's a score of joint and osteoarthritis and things like that, and again, females kind of dominated the improvements when it came to use of rapamycin, and not so much with male patients. For SF-36, higher is better. For WOMAC, lower is better. All right. So where does that leave us? I mean, what can we kind of take away from this, and what are the next steps? So as we talked about, there didn't seem to be any significant difference in adverse reactions, events, or there were no concerning biomarkers. Like we didn't see anybody's liver functions go sky high, or cause creatinine to go high as well. So at least within kind of this limited timeline and limited study, we didn't see any reason to be concerned about patient safety. And it does seem like, the low dose weekly rapamycin, at least over a year, had signals towards improvements in a few health domains that we looked at. And then, like I said, there is variability here. So, any work that needs to be done, I think from here kind of has to figure out what's behind the variability and how to address it. I suspect that, if you find the right dose for the right individual, they would respond. Although there could be a certain percent of people, for whatever reason, may not respond, no matter if you find the right dose or not. We see that with other therapies as well. But I think that's just my suspicion, my clinical suspicion here. It definitely needs to be followed up with more. So next steps, I mean, kind of what are we doing? We like I said, we did a evaluation of biomarkers, looking at sirolimus levels, at a 24-hour measurement. And like I said, we published that data. We did a whole webinar. You can look at our you can look at our social media channels to find that webinar to understand what we did. But we're continuing to follow up on that. We now have hundreds of sirolimus levels on patients on rapamycin across various different time points. So at some point we'll be able to start putting together graphs and charts and things like that, and maybe be able to derive some insights there. Now, I do have to warn you that it's possible that levels, the sirolimus levels are not the key. We're not sure. It's possible it could be. It's possible it could not be. I mean, it's possible you could have same sirolimus levels in two different people, but they respond differently to the same sirolimus levels. So we're cautiously optimistic, but it's possible there could be something else going on. Genetic variability, lifestyle, microbiome, on and on and on. A lot of variables here, but you got to start somewhere. And then, like I said, we are using this study to kind of continue. A lot of the participants from Pearl went on to be prescribed rapamycin either through us or through other clinics or services. So we are hoping to collect more longitudinal data on those participants as well as any new participants. As an observational, it's not going to be an RCT, but I think it'll be a larger data set, much larger than I think what we could do in an RCT setting. We were able to biobank some samples from our participants, but the logistics of the biobank turned out to be way too difficult, which was one of the many limitations. So we only had a limited amount of biobank samples, but in the future we hope to be able to have more biobank samples across a larger set longitudinally. And one of the reasons I personally got interested in rapamycin was I've heard several reports from rapamycin users and even doctors who prescribe rapamycin that people will report either weight loss, like a mild weight loss, like modest, or or they're able to keep their weight maintained better. And so we're kind of looking to see whether it's one of the tools we could potentially use for people coming off of GLP-1 medications, once they've hit their plateau or once they've hit their goal. Some people may not want to be on GLP-1 medications for a long time. So we are initiating a study looking at kind of if rapamycin, along with other we have the study set out along with other therapies, looking to see whether it's part of the solution for weight loss maintenance in the future. Okay, so of course, this was a community effort. A lot of people came together, to help us out. We had advisors, we had the IRB. We had our researchers, we had our staff, we had, of course, Lifespan, we had our donors, we had our participants. There's so many people to thank here. So, for everybody out there who supported us or, attended our webinars throughout the years, thank you very much. And we look forward to expand these efforts and then, of course, look at combination therapies, because no matter how powerful rapamycin may or may not turn out to be, it's just one tool. And I don't think we can expect to conquer aging using the one tool. It definitely needs to be a combination effort. Okay, we can open it up for Q&A.
Fantastic. Thank you so much. And I think a good way to do this is perhaps we can go through some of these questions and open up the floor to the panelists, including yourself, Dr. Z, Matt K, and Oliver. And if you guys want to give your thoughts on the trial data as well before you answer the first question, that's be great as well. So, I'll start with one of the questions being, you know, why did we choose compounded rapamycin when going into this trial? And given the results at hand, do we think there are any advantages of compounded rapamycin, either logistically or, biologically?
Oh boy, how much time do I have to answer this question? Yeah, so again, going back 5 years, compounding medications tends to be a favorite among like integrative medicine doctors, functional medicine doctors, longevity medicine doctors, so that was like one of the first reasons why was that just kind of seemed to be a trend, and we wanted this study to be as practically applicable as possible. So that's part of what drove it, was kind of the practical applicability. The commercially available doses are very limited. You got 0.5, 1, and 2 milligrams, and that's it. Whereas compounded, you can ask the compounding pharmacy to adjust it to whatever dose you want. And that kind of would have potentially set the stage for then customized dosing, et cetera, et cetera. So one was a kind of a practical aspect, like what would prescribing look like after Pearl? The other aspect, and this is, I think, underappreciated, is the placebo. So rapamycin, if anybody's seen the Rapamune or the Dr. Reedy generic tablets, they have this weird pyramid shape with the smooth coating on it. And finding a placebo to match that to be like a truly randomized, like blinded trial was nearly impossible. It would have like dwarfed our budget like multifold. And plus we would have had to have then 2 placebo groups, at that point. So there was a practicality issue. We didn't know. We didn't know at the time. It was never like brought to our attention by any of the advisors or any of the articles that reviewed that there was a potential difference in bioavailability. It was only about, like I said, 1/3 of the way into enrollment that I think credit goes, I think, to Ross Pelton, who brought it out, that along the way rapamycin became encapsulated to improve its bioavailability, and then we later found out that the ITP had trouble with absorption with kind of non-modified unmodified rapamycin. So we actually paused enrollment and we did like some preliminary data, and we enrolled a few volunteers and kind of did a head-to-head comparison, and there was clues there that there was bioavailability, it was just lower. And then the larger study that we published seemed to indicate, there was like this 1-to-3 ratio of absorption. So then we had to make a decision. Do we, stop the trial and start all over again with a higher dose? Or do we just kind of continue and hope that the lower availability, will give us some results? And I think ultimately, hopefully, we've made the right choice of continuing on. Otherwise, we wouldn't be having this webinar today. But that does kind of highlight a limitation. Now, the benefit, and this is only speculative, is a lower bioavailability means more gets into the gut. And so it's possible that could have an advantage. We don't know.
Yeah. Matt, do you want to give your thoughts on that?
Sure. So a few thoughts. I'll start with the last speculation. That's possible. My guess is probably not, given that rapamycin is unstable at gastric pH. So my guess is what actually happens is you have less get into the small intestine because it just gets broken down. But I think we don't know is probably the honest answer. So a few things that I would say. I think this issue of compounding points out one of the challenges of doing clinical trials in general, and particularly these kinds of clinical trials, is you have to take your best guess on a bunch of stuff when you start, and then you sort of have to live with it for a variety of reasons. And those best guesses are hopefully mostly driven by science and what you know of the literature. Sometimes they're driven by pragmatism and what you can afford or what the regulatory bodies will allow you to do. So I think that there's no reason. I don't think there's any reason to be critical of the team that designed the trial over this compounding question. It was the best information they had at the time, and they went with it. I think maybe the other thing that I would say is I can just kind of give my take homes from this study. And before I do that, it's probably worth saying that I wasn't involved in the design. I don't have any financial interest in AgelessRx. I know some people think that I have some sort of financial interest in rapamycin. I don't. I'm not going to make any money if rapamycin suddenly becomes the drug that everybody wants. So my take homes are, first of all, I want to say, Dr. Z, I think did a good job of presenting this in a balanced way and not overstating the results. I think the strong conclusions we can draw are pretty limited, but there are a few things we can say. I think one, this fits into a larger body of evidence that rapamycin across a pretty wide dose range, I think we're learning, can be used safely in otherwise relatively healthy people. So outside of the context of organ transplant patients, taking other immunosuppressants and taking high doses of rapamycin, usually daily, I think we're seeing that that rapamycin is pretty safe in the doses that people are thinking about using off-label. So this is additional evidence over a longer period of time, which I think is always helpful to have additional safety data. I kind of think the one piece of data here that jumps out to me are these changes in lean mass. They look real to me, and they're at least statistically significant in the women. That also fits anecdotally with what my experience has been and what other people have told me. And I think what Dr. Z said about the weight loss is the same thing that I have been told by multiple people taking rapamycin and physicians who prescribe it. So I think there's real biology there. I think we can even point to some potential mechanisms based on the literature. I believe that's probably real. And so I think it's nice to see some evidence supporting that coming out of, what is admittedly a limited study with probably suboptimal doses if we were to go back and design it from the beginning. The last thing I'll say is I know a lot of people will be frustrated by the fact that we're saying it's limited what we can interpret from this study and we need to do more studies. I don't know what else to tell you other than deal with it. If the people who can and should fund the larger trials for efficacy would get off their asses and step up and do it, we'd be able to answer these questions definitively. But there's no point in complaining until somebody is going to step up and fund definitive clinical trials on rapamycin. It is an ongoing source of frustration to us all. We get it. Again, the funders have got to step up and do what they should have done 15 years ago and fund these trials.
Great. Well, thank you for that. Appreciate your input on that. Anybody else have any, Keith or Oliver, any?
Yeah, got a quick question. This you might not be able to answer this given the limitations of the trial and the size of the amount of participants. Given what we've seen from the data here, you've got differences in kind of who's improved. It's sort of like there's some variability there, and I know there were some male and female differences, but you had a kind of broad age range from 50 to 80. So I'm just wondering, given the data that you have, is it possible to figure out if there's any potential age category differences in the responses to rapamycin here, like between the 50 and 60 group and the 70 and 80 group, for example? And would that account for any of the variability, or is the data pool too small to basically determine if that's operating here?
Yeah, I think Stephanie would be able to answer because I know she sliced the data, sliced and diced the data every which way to kind of look for any kind of patterns.
Yeah, I worked with a biostatistician that collaborates with us a lot, and we both, in different ways divided the data every way imaginable, reasonable, unreasonable, just very exploratory in some
Well, yeah, I just wanted to to piggyback on one of the other things that Matt had said, which is that of the need for funding, right? One of the things that I was grateful to see as a result of doing this was, as some of you may know, we ran a campaign on lifespan.io back in 2021 to raise funding for this, and this project was by far the largest amount that we raised via crowdfunding from over 400 people. I think it was almost 200,000 on the campaign itself, and then there was some additional funding that came in sort of parallel to that, which is, on the order of like, a good SBIR grant or something like that. So I think it presents this alternative model that can be considered more and has this additional ability to kind of rally the crowd, become very publicly visible, get people informed and engaged. So I just want to just like sort of paint that target that, if there are additional follow-up studies that seem like they need to get done, that's something that we'd like to collaborate on. And, to go to what Matt said about people getting off their butts and funding, I think part of that is on us too, to kind of like get it out there into the world, because one of the frustrating things for me, I'm sure it's for Matt and everybody else on this call, is like there are a lot of questions like this one, like what is a safe dose for rapamycin in humans? Like going back since like 2006, people have been asking this question. It's like a ready-made, like someone should investigate that. So I think we need to really get these kind of questions where everyone kind of agrees that this is a good thing. We need to do it and get it out there to the public and say, hey, let's answer this question and you can be a part of it. So again, all caveats, of course, that, still more work to be done. There's only so much that you can draw in terms of the conclusions from this particular study. I'm very optimistic that, as Matt was saying, that if we had more funding going to this, we can really definitively answer these questions in a way that might, give us another potential therapy that regular people can add to their regimen. So
yeah, one thing that really, I think will help accelerate progress in these studies is the X Prize. I don't know like how much that's been discussed in the community, but I think there's a lot of interest in expanding or exploring these therapies, the various different endpoints and things like that. I wouldn't be surprised if multiple teams included rapamycin in their in their regimens. AgelessRx hasn't decided whether we're going to throw our hat in the ring or not, but we definitely are interested in doing so. And I think rapamycin, seems to hit at least 2, possibly even 3 of the domains that they're testing. So I think that could be a catalyst, or one of the catalysts that we need. But of course, it never hurts to have more.
Yeah, it's worth noting on that I believe combination therapies can apply as well. So maybe if rapamycin is paired with something else, you can hit all 3 domains. So that could be interesting. Yeah.
Oh, I already have ideas. Yeah, we're already putting together a proposal, but like I said, we're not sure if we're going to enter the race ourselves or as part of a larger team. The intention's there, whether or not we have the resources and things like that to actually be a strong contender. We're still evaluating. But if anybody is interested, you could reach out to us, and like I said, never hurts to have partners or team up on these sorts of things.
Thanks for that, everyone. And I just want to reiterate after this discussion from the panelists that what we saw from this study, right, leading into the next question, is in terms of overall effects between arms, 5 milligram, 10 milligram, and placebo, we didn't see any statistically significant differences, right? But what we did see, which we had emphasized before, Dr. Z, Matt, is a statistically significant improvement in lean muscle mass, albeit a smaller size, right, in women specifically, with 6 individuals, right? And other robust results from the study were an improvement in self-reported pain within the SF-36, as well as statistically significant improvements in WOMAC pain, overall WOMAC score, which is an osteoarthritis scale, and overall quality of life. So these are the things that we cast the net broadly. We saw gender-specific effects, mostly within woman. And this could be because of the lower blood levels we were getting. Now, we didn't measure blood levels, but from our rapamycin bioavailability study, we saw about a 1/3 ratio. So it could be that higher doses are more effective in men. So I wanted to reiterate all this just leading up to the next question, because I think it's important. I'd like to get everyone's perspective here, because we're talking about funding trials, we're talking about getting attention, but if we were to use this trial as information to inform future studies, knowing what we know from these results with what I'm going to call, and I'll give credit to Krista Copy during a discussion at ARDD for this, very low rapamycin or very low dose rapamycin. How would we redesign the study or implement the next one in terms of dosing and in terms of primary endpoints, right? I mean, I think the mind immediately goes to immune system and, based on John Manick's studies, but do we really have good markers for chronic inflammation? Outside of body composition, are there any other endpoints that we would focus on that we think, there's a there, and what doses will we go for? And I'd really like to, go through the panelists and get your thoughts on this.
Sure. I'll start. So actually, just to jump on that chronic pain comment that you made, just worth mentioning in the survey-based study that we published in 2023, that was one of the differences that was seen between the rapamycin users and the non-users was in the rapamycin users, self-reported fewer or lower levels of chronic pain in that group. So again, I think when you start to see multiple arrows all pointing towards the same thing, you can start to have a little bit more confidence, even if none of the studies are done perfectly the way you might want to do them if you had unlimited resources. So it fits. So in terms of endpoints, again, I think rapamycin is interesting because you have kind of your pick of a lot of different stuff that goes wrong during aging. So again, based on this study, I think I would be more inclined to include a body composition analysis, maybe paired with resistance training. And I think you can ask questions around dose. Actually, I think this study is valuable in the sense that everybody's sort of like, well, the dose was too low. We didn't know that going in, right? Everybody's making that assumption that 6 milligrams once a week is this, exactly the right dose off of basically almost no data. So, I think we've learned, probably threefold lower than that, which is what we're guessing is about what these people experienced, is probably lower than what you would want to do if you were going to do the trial again. So you'd probably want to start with something in the 6 milligrams once a week range, but I could see a rationale for trying to go higher than that. Again, you'd need to establish safety at the higher doses. So I think there is value in the way this study turned out, though, because we learned that, this lower dose is probably suboptimal. And maybe there is a sex effect in people like what has been seen in mice, that women may be more sensitive to rapamycin. So maybe you would want to design a trial that looks at a lower dose in females compared to males. Okay, so now your question about endpoints. So I think about body composition for sure, for this study and other information that I've heard and experienced anecdotally. Obviously, if you know me and my research, I'm a big proponent of oral health. I think that's where we see in mice huge effects from rapamycin. The endpoints are non-invasive, easy to measure, inexpensive. There is a small clinical trial for periodontal disease happening right now, so I think about that. At menopause is interesting. Again, as many people on this call probably know, there's a clinical trial out of Columbia, Zev Williams and Yushin Su, looking at premature ovarian failure. There's a bunch of anecdotal stories about perimenopausal women taking rapamycin who experience interesting effects. I would think about looking in that population and looking at hormone levels, symptoms of menopause, things like that. So I think that's a very intriguing area to think about for clinical trials. Cognitive function, tons of data in the animal literature on improvements in age-related cognitive decline and dementia. So that's another place you would obviously think about looking. Measurements of senescence in skin, maybe. Again, there's a couple of studies showing that senescent cell burden in skin goes down with rapamycin treatment. Again, pretty easy to do in a clinical trial. So I think you just have, and I could go on and on and on. I know I've talked for too long. This is one of the challenges. You've got a bunch of stuff you could look at. You have to pick, and I think where we need to spend a little bit more time as a field thinking about I think almost all of the geroscience clinical trials today are not powered for efficacy, period, full stop. People don't recognize this. You look at all the senescence trials, they're safety trials. So I think we need to start to move towards efficacy trials and stop doing underpowered trials where showing that there are limited side effects is called a win. And so I would like to see a move towards picking endpoints, doing statistically justified clinical trials for efficacy so we can start to get to some answers.
Thanks so much for that. And I see, Keith, you have your hand raised.
Yeah, I just had a quick follow-up question based on some of what was just said. This might be tangential to the geroscience angle, but I'm guessing the answer is no, but has there been any studies investigating potential effects on from rapamycin on those with musculoskeletal issues, your palsies and these kind of things, to see if it remediates pain, since remediation of musculoskeletal pain seems to be something that's coming out of data like this? Do we know if any of that work has been done?
There is a study. There's a study. I think it's just going underway in New Zealand about muscle function by Brad Stanfield.
Yeah.
I don't know if he's looking. I mean, you guys helped him crowdfund for it. I don't think he's looking at pain, but I think it wouldn't be unreasonable to add, to evaluate to see what kind of pain response to rapamycin. Is it like bone-on-bone osteoarthritis pain? Maybe, maybe not. Is it like, itises like tendonitis, bursitis, things like that? Probably. Would fibromyalgia type pain, or like non-specific or neuropathic type pain? Maybe. I think these are all very good questions, and I haven't seen any formal data on it. We are looking at like I said, larger observational longitudinal data points through our telehealth platform, and maybe we'll get insights. So ask me again in about a year.
Yeah, I was just thinking that could be another big inroad for higher, for example, if this could be a remediation of arthritic pain or something like that, you could see that explode usage of this. Yeah.
So Keith, I always hesitate to say there has not been a study when it comes to mTOR or rapamycin, because you look on PubMed, there's a study for just about everything connecting to mTOR, but I'm not aware. But I will say there's quite a bit of anecdotal evidence out there, people self-reporting this, and there are some people using rapamycin in combination with ketamine for chronic pain and severe chronic depression. So there are interesting connections outside of the aging space for rapamycin having impacts on pain and depression. Is that brain mediated, or is it more, at the site of the chronic pain? I don't think we really know it at this point. So shameless plug. I haven't mentioned the podcast, the Optispan podcast, but I'm actually going to be interviewing a psychiatrist who uses rapamycin in this way in the next couple of weeks, and that'll be on the podcast. So I think and I should also mention, in both of our short-term clinical trials in dogs, again, double-blind, placebo-controlled, the owners self-reported that these are old dogs, that the dogs were more active, which I think one reasonable interpretation of that is a decline in chronic pain that a lot of old dogs experience. So again, I think there's arrows pointing in the right direction, and we just really need some definitive studies to answer that question.
Yeah, good points. And I think, if I'm not mistaken, I might have come across a paper from the Marmoset study, Adam Salmon, where he looked at bone-related osteoarthritis, and there were some improvements there. So there are several signals pointing there. So definitely an area worth diving a little bit deeper into. Another question that came in quite a few times was this push towards measuring blood rapamycin levels and the value that could bring to clinical trials, to clinicians, as well as, are there any robust biomarkers of mTOR activity? And if we were able to converge on measuring blood levels and/or biomarkers of mTOR activity, how do you think that could affect clinical outcomes? And I know likely be a lot of conjecture here, but interested in thoughts from the panelists. And yeah, Matt, if you want to start off.
I can jump in again. I'm good to speculate. Well, I speculate a lot. I'm not sure if I'm good at it. So again, shameless plug for the podcast. We've actually covered this topic in a few different episodes, so I'd encourage people to check that out. I think we don't know, obviously, what the optimal levels of rapamycin are, either in terms of dosing or blood levels. I think what we do know is there's a bunch of individual variation, both in the bioavailability of a given dose and the rate at which that is cleared. And so that variation, I think, really complicates our assessment of finding an optimal dose, because again, it's a multivariable sort of equation that we're trying to solve for. And we don't actually know what the endpoint we're solving for is. Like, this gets to your question of how do we measure mTOR? And it's going to be different in different tissues. I think that's part of what also makes this challenging. Even if we had a really good biochemical assay for measuring mTOR activity in blood, the mTOR activity in the brain from a rapamycin dose is going to be different than it is in the liver, is going to be different than it is in the kidney, the intestine. You can go to all the different tissues and organs. So I just don't think we know enough about what the optimal thing to measure is to get to an optimal dose that way right now. I know it's not satisfying. I don't have an answer, other than that's why I think what we really need are the functional endpoints that we can pair with the molecular endpoints. And again, I think you guys have started to try to do this a little bit. I'm very wary of this responder, non-responder analysis, as Jurish knows, because I told him. But having said that, I do think you're trying to figure out, like, can we correlate microbiome changes or LDL or other blood markers with what we think is a signal, functional signal for response or non-response to rapamycin. I think those are the kinds of things we need to get better data on to be able to try to optimize dose. Because right now, there's so much speculation in this idea of, weekly dosing, knock down mTOR complex 1, don't affect mTOR complex 2. We don't even really know how to measure that. I'm just not super confident trying to go that route to say you should take 6 milligrams once a week and this other person should take 12 milligrams once a week. I just don't think we know. So we're sort of doing the best we can, a little bit stumbling around in the dark. Sorry.
It would be, it would be really nice if there was some, whether it's just through the regular biomarkers, like, some changes in some blood marker, let's say RDW or something like that.
Yeah.
That would help indicate, or a combination, or something more anatomical, like, for example, visceral fat, for example.
Yep.
Let's just hypothetical. So let's say, you dose your rapamycin until you see a drop in your visceral fat, for example. That would be really nice to have. I think that would warrant further studies to see what are these kind of surrogate markers we could look at. When this is kind of off topic, but when we sponsored a low-dose naltrexone trial on mice at the University of Washington, and John N there, he said that weight loss was one of the indicators for the mice. So something like that, where it's working because XYZ. Canker sores is not a good sign. Some people use that to say, oh, I'm on right dose or not right dose, because I think only about 20% of people actually get the canker sores. It could be, though. It's possible. I think at this point anything is possible. I have had a couple patients where the dose, they got up to 6 milligrams of rapamycin. The canker sores were too intense. You knock them down to 4, and it seems to be a lot more manageable. So it's possible 6 might be overshooting. So I'm hopeful that we might be able to find something simple like that, but maybe I'm just being too hopeful.
Yeah. Can I just add one more thing that occurred to me when you were saying that? I think getting more data on blood levels will be useful, sort of, irregardless of the fact we can't necessarily translate that to mTOR inhibition in all the different tissues. And I think one reasonable place to start is a hypothesis, and it's a hypothesis because we don't have data to support it really yet. But that if we really believe once weekly dosing is a good strategy for the reasons that people talk about with mTORC1 and mTORC2 and others, you could think about a strategy where you dose and you make sure you get below the detection limit of the assay before the next dose, right? And you can measure that. And for some people, it might be 6 mgs. For some people, it might be 12 mgs. For some people, it might be 20 mgs. And then try to correlate that with side effects like mouth sores or other things you can measure like RDW or other blood-based biomarkers. The other thing I'll say is one of the tests we use as at Optispan is an immune functional test from Superior Bio that we think may give us some predictive capability for responders and non-responders to rapamycin, at least in terms of what that test measures. So I think there are some opportunities down the road to look at immune-based biomarkers, which probably are going to be the most sensitive to rapamycin, given what we know that how we know rapamycin works.
Yeah, there is just on that RDW MCV, we not in the Pearl data, but when I've been kind of looking at some of our more longitudinal data from our telehealth platform, there is this phenomenon that I'm seeing, not in everybody, but in a few people, where the A1C seems to jump up, but that does not correspond to an increase in glucose. And I'm wondering whether there's something to do with the turnover rate of red blood cells. So I'm wondering, if that kind of phenomenon needs to be studied more closely, and see if there's some correlation there between these potentially bone marrow changes, or could be a splenic change, or stabilization of the red blood cell matrix. I don't know. It's but it is I've seen it over and over again in multiple people. Again, and I'm kind of scratching my head as like, what's going on? Why is A1C jumping up by usually 0.5%, which is big. But you're like, oh my God, what are you doing to this person's blood sugar? But you look at their fasting blood sugar and it's the same. So that is something to kind of look at a little bit deeper in the future.
Yeah, those are all great points, and I think Dudley Lamming is organizing a clinical trial now in the works where I think it's A1C that's one of the primary endpoints. Matt, I guess someone can correct me if I'm wrong on that, but there's a lot of there's a lot of different targets throwing the dart at that dartboard in terms of what these effects can be. Another question that we got a lot of, and I think people are really enthusiastic about this, and I think it's important to address perhaps again, is perhaps why didn't we include biological age tests, DNA methylation clocks, for instance, as a primary endpoint, or at least elevated as a higher impact endpoint. And I think it's worth discussing thoughts on that a little bit further.
Yeah, I mean, we did. We tried. We had a partner, Glycanage, and we had True Diagnostic. Although I don't know if we had enough people do the True Diagnostic to make enough sense of the data. But I think Glycanage, I don't remember what their full analysis was, but I'm not sure if there was any major changes there to kind of speak of.
No, I can speak to that a little bit. We only had a subset of, we already had a fairly small cohort, and then only a subset of those opted into the additional testing. So we just didn't have enough samples to really see significant differences. The biggest differences we saw were by gender, which were not very surprising. You see differences in methylation patterns by gender, no kidding. So it's something that we would love to figure out a way to get more people to participate in the future so that we can make stronger conclusions. Yeah. I think that's a take-home message for everything. We would love to have more participation.
Yeah, I'm not optimistic about seeing methylation changes, not with the current clocks anyway. I've heard from multiple people, including Steve Horvath, he kind of told me once that, hey, look, I've looked at unpublished data on rapamycin and methylation clock, and it doesn't seem to be any signal there, but that could also speak to the severe limitation of methylation clocks. Is like, well, RAP is doing something, the dog trials, the mice trials, the human trials with, gingivitis getting better and tendinitis getting better. We know it's doing something. So it might just be, it's just not doing the right something to show up in these methylation clocks.
Yeah, and can I just add? I mean, I think nobody serious would use a biological age clock today as a primary endpoint for a clinical trial. They're just not ready for prime time in that way. I think they're good research tools. I think you could do it as an exploratory endpoint, and that sounds like what you did, and that makes perfect sense. My concern, though, would be we don't really know and again, look, we use Glycanage and True Diagnostics at Optispan as part of our exploratory analyses that we do on some of our clients. So I'm not bashing the companies. I'm just saying we don't know anything about variants, technical replicates, that is all opaque to us. So it is not an appropriate type of data for a rigorous scientific clinical trial without that information. It's hard to know how sensitive these tests are. You can't do any sort of statistical power calculation without having that information. And so I feel like it's fine as an exploratory thing, but I think a lot of people watching this webinar really don't understand some of the limitations with these biological age tests as they exist today, and especially using a for-profit commercial company as the vendor for trying to do a clinical trial using these tests. Sorry, guys. I know there's probably some people from these companies listening. Again, not trying to bash them at all. Great companies, great people, but there are some limitations to the data that we've got about those tests.
Which highlights that we need, multi-omics. We can't just rely on a methylation clock or something like that. We need data from like to aggregate the data and come up with some sort of, I don't know, master aging clock. If there ever is going to be such a thing, it's just too early to say, I think.
Like two other ways they're being used real quick. There's one trial in Singapore that is using, I think it's DNA methylation clocks as a screening tool for inclusion, exclusion. If there is, I mean, like when you look at these biological age clocks, I think we definitely put too much weight on them, but there does seem to be associations that you can make, right? Are they as good as looking at hemoglobin A1C or other clinical markers? That's an open question. But I just wanted to put out that there may be other ways to use them as well that aren't tied to efficacy per se. And especially with these omics tools, if you can find patterns, again, I wonder if you can use them to predict clinical effectiveness if they're stable enough. That's the big question, stable across time.
Yeah, I think you can, although again, I think we want to be careful about who's doing the measuring, where is the measuring being done, and what do we know about the quality control at the organizations where these measurements are being done. And again, there's no reason to think that a for-profit company can't do as good or better than an academic lab at doing these assays. I am sure they can, but we don't have that information. That's what I'm saying. We don't have any information, as far as I know, about the quality control at all of these different companies that are offering these epigenetic tests to consumers. So we just don't know. And why would you believe it in the absence of that data? As a scientist, that's the question I always have to ask.
Absolutely. All right, so I just want to, before going forward, perhaps I just want to acknowledge that we're 15 minutes over the hour. I know that previously the panelists had said that you guys are willing to go on. If that still holds true, that would be great. I still see we have about 150, almost 150 people in the room, so we could keep going if if you guys are willing. I just wanted to do a quick check on time before going further. Yeah, thumbs up.
Yeah. Maybe another 10, 15 minutes if everybody's okay with that. We can probably knock off a few questions. Yeah. So Grish, what's up next in terms of like themes of the questions?
Yeah. I did want to, so one of the questions were, and this may have been addressed in one way or another, but sexual dimorphism, these sex-specific effects have kind of been popping up in the literature with rapamycin, right? With mice, you see enhanced lifespan compared to female mice compared to male mice, a higher bioavailability. In marmosets, it's kind of the inverse relationship where it seems that there's a higher bioavailability in male marmosets compared to female. And again, talking about standardization, it really does matter when we're measuring rapamycin levels. Is it at a peak? Is it a trough? There's a lot of complexity there. Within our bioavailability study, we did not see any differences at the 24-hour mark, 24 hours after taking generic or compounded rapamycin. We didn't see any differences between men and women in blood levels. Within the Pearl trial, we do see some sex-specific effects, statistically significant sex-specific, female-specific effects. Can you guys talk to or address what you think may be driving some of this based on what we know at hand and perhaps even some hypotheses you may have?
Well, first of all, the question I think was asked, why did or didn't we check sirolimus levels during Pearl? We started out actually, so this is maybe a clinical Pearl for people who are doing studies, but it turned out we could not prevent people from seeing their sirolimus levels. So we were in danger of unblinding the participants. So we had to stop checking sirolimus levels on our participants. So we purposely stopped checking sirolimus levels. So that's one unfortunate thing that we learned after we launched the trial. I think it could be something with the immune system. If you think about, females in general tend to be much more prone to autoimmune conditions, for example. Things like fibromyalgia, which is not classically an autoimmune condition, but thought to have some kind of immune dysfunction to it. So it could be that, rapamycin is working in a way that improves their immune system, that men just don't have that same issue with, for example. But that's just purely speculation.
Yeah, and from my perspective, I mean, I think first comment on the mouse data, there's this sort of misperception that rapamycin works better in female mice than in male mice. As I think the data on whole don't support that. What the data on whole in mice suggests is that at a given dose, female mice are either more responsive or have greater bioavailability of rapamycin. So the dose response is shifted, but if you keep pushing the dose higher, eventually the male mice will get the same benefit that the maximal benefit seems to be in female mice. So that, again, that hasn't been completely worked out, but I think that's pretty solidly what the data suggests right now. It's not that females respond better. It's a shift in the dose response. Okay. In humans, I don't think we have any evidence that same phenomenon is happening. I think Dr. Z's speculation, it makes perfect sense. It could all come down to chronic inflammation and immune-related processes where in females, those are just more of a burden at a given stage of life than in men. I do wonder whether this is at all related to perimenopause and menopause. This has already come up once, given the evidence that rapamycin can affect ovarian aging, can potentially affect symptoms of menopause. That may be tied together. Those are not mutually exclusive, and probably some of the inflammatory symptoms are driven by hormonal changes. So I think those are connected and reasonable potential explanations for what was seen here.
Yeah, excellent. And just as a note, the average age within our study arms within the Pearl trial was around 60 to 62. So postmenopausal woman. So that's definitely interesting. Another question that came in, and is the field seems to be focusing in on rapamycin / sirolimus quite a bit in terms of at least the longevity field, the longevity community, right? And this is in part because there's really no other affordable and accessible options. Joe Manick did test everolimus. Do you think that sirolimus is the optimal I don't want to call it a rapalog, it's their OG, but is it the optimal form, or are there other rapalogs in the pipeline that are like everolimus that seem to be more promising? And I know there some of this is contingent upon this hypothesis, largely built by Dudley Lamming and crew of more specific mTOR1 targeting is going to lead to greater efficacy and reduced side effects. But interested in both your thoughts on that.
I'll take the practical aspect of it, and Matt can talk about the scientific aspect. So everolimus is incredibly expensive. I think when I looked it up, if you look at GoodRx or something like that, I think it's like 10 times more expensive than rapamycin, sirolimus. And rapamycin itself is not an inexpensive medication either. So that's one practical aspect. And from my perspective, I mean, I don't think there are very many animal studies. So I just think we know even less about something like everolimus than rapamycin or sirolimus.
Yeah, I agree completely. I think right now, rapamycin or sirolimus, which for those of you who aren't rapamycin aficionados, those are the same thing, two different names. That's the best game in town, given the data we've got. I also think it is, to the extent that I can be 100% certain about anything as a scientist, I'm 100% certain there are better mTOR inhibitors out there in terms of efficacy. We just haven't looked, honestly. And Dr. Z is absolutely correct. There's a little bit of data on everolimus almost nothing on other mTOR inhibitors in the basic literature for longevity. So we just don't know because nobody's looked. I can tell you a company we spun out of my lab called Aura Biomedical, which is a high throughput longevity drug discovery company, primarily using C. elegans, looked at only a few dozen mTOR inhibitors and found at least one that's much better than rapamycin for longevity in C. elegans. So I think if people were to go look, we would absolutely find drugs that target mTOR and have better efficacy than rapamycin. Would they be rapalogs? I think is an interesting question. So again, for people who aren't in the weeds here, rapalogs you can just think of as derivatives of rapamycin that biochemically inhibit mTOR through the same mechanism as rapamycin. There are other ways to inhibit mTOR biochemically, and drugs that do that. Again, nobody has really looked at these other types of mTOR inhibitors in any great depth in the context of longevity. So I am pretty confident that there are things out there better than rapamycin in that context, but nobody has looked. And rapamycin works pretty well, we think. So, I think pragmatically, that's the thing that we should be focusing on right now.
Yeah, we can't get people to fund rapamycin studies, these other mTOR studies. Yeah.
And I do think this question of mTOR complex 1 specific rapalogs that you mentioned is an interesting hypothesis. It is still a hypothesis. It has not been proven that you can get better effects on longevity or health span with a rapalog that is more specific for mTORC1 and less off-target effects chronically on mTORC2. So it sort of gets talked about as if that's true. That has not been proven. So somebody needs to actually show with real data that an mTORC1 specific rapalog is better than rapamycin, I think, before we should get too far down that road and really believing that's the case. There are companies developing these drugs. Hopefully we'll see some data sometime in the near future to support that hypothesis.
All right. I think we take a couple more questions, I think, before we have to wrap up.
Sure. I'll lead off with a more of a fun one here. Being totally speculative, how large an impact does the panel think rapamycin could have with the most optimistic, hat on in terms of X years health span or Y years lifespan? Based on the data at hand, based on what you see, we see within trials that lifestyle change can have anywhere between the 10-15 year extension of healthy longevity. How do you think rapamycin compares right now or could compare once we start optimizing and standardizing protocols moving forward?
I think Matt's the better one to answer that.
Well, this is pure speculation, so we're all entitled to our opinions,
please,
right?
Yes.
So again, I think it depends on who you're talking about, right? So if you're talking about somebody who's got the other factors more or less dialed in, doing a reasonably good job on lifestyle factors, I almost hesitate to speculate on this because I know it's going to end up on X. Kaeberlein says whatever I say. I feel like, realistically, for some people, rapamycin can probably get you a decade of quality health span. Total speculation, but I think that's, given what I know and what I have experienced, I think that's probably a reasonable speculation. It might be more, could very well be less. For some people, it will be more in terms of quality of life, because I think particularly people who have a high burden of sterile inflammation, which can have a pretty high impact on quality of life. I think rapamycin is very effective in some people at knocking that down. And that could be a really, really large impact on quality of life for those people for many, many years. So it could be bigger than that for some people. But I think probably if you've got most of the other stuff dialed in, it's not going to be 30 years. I don't think. I hope it is, but I don't see anything that's making me believe that at this point.
Yeah, I mean, I think I like to be optimistic about it, and I think if it is making some fundamental improvements to some of the hallmarks of aging, like inflammation, like microbiome, things like that. I mean, you could see a scenario where over time you see a bigger and bigger improvement between the users and the non-users. How far that'll take us, I don't know. I mean, even I think you covered this on your recent Optispan podcast, but even with some of the more sophisticated genetics and manipulation and things like that, with mice, they seem to hit a wall. They keep going,
Yeah.
and then there seems to be a wall, like no matter how well you do on the health span aspect, and then and then they kind of fall off a cliff. Humans could be different. We are different in many, many ways, so it's possible. Humans could go past that wall. Rapamycin by itself, probably not. I think you definitely have to add other therapies to rapamycin if you want to have the best shot.
Keith, you have your hand raised.
Yeah, I was just going to jump in. Obviously, I'm going to defer more to the true biologists among us on these issues. But yeah, just from what I know, I echo what Matt said in that I would be surprised if it went beyond something like 10 years. I would think of that as an upper bound. But something I wanted to venture forth as something I'm excited about that, further testing on rapamycin can sort of add to the overall, soup of knowledge here is that one question that I think is not yet been sort of sufficiently disaggregated is if you do get a life extension benefit or a health span benefit, how much of it is due to some core systemic things that are actually changing versus pain remediation, these sort of secondary factors that might make you become more active and then get benefits that way. So because it seems like rapamycin has those potential positive side effects, I think in the future, it will be important in those studies later to disaggregate what are the mechanisms of actual health span extension. So that's one of the reasons why I'm excited by the results here, actually, because I think that'll be, because, at the end of the day, we, of course, want to get to the root of the things that will be changing something fundamental and core and are not only getting benefits on those like secondary, tertiary things. But I think whether this has that as well, I can see this as being important in that grand hypothesis testing later. Does that make sense?
Yeah, I think that's a good point, Keith. I think the one thing that would make me suggest that I think we already know rapamycin is doing more than that, at least based on the animal studies, because as Dr. C alluded to, we can find evidence in the literature that all of the hallmarks of aging are impacted by rapamycin, and we actually know some of the molecular mechanisms there. So I think it is clearly the case, again, at least in laboratory animals, that rapamycin is modulating biological aging as we understand it today, right? So it's not only improving activity indirectly. It may be doing that as well. And I agree with you. We need to understand those mechanisms and appreciate what the relative contribution is. But, and I only say this because I think sometimes people get confused and they're like, well, rapamycin just prevents cancer. And I'm like, well, okay, yeah, it delays cancer, but it does all this other stuff too. And we know that. I don't think we can, I think we can be sure it's not only doing one indirect thing, and that accounts for all of the lifespan and health span benefits that have been seen in laboratory animals. Of course, people are different and we don't know yet in humans.
Yeah, awesome answers, and I asked everybody to wear their optimistic hat, but I still think that the oldest woman alive just recently died. I think pretty recently, at 117 years old. She was from Spain, and we have case examples of individuals living relatively healthy lives, centenarians. So extending that health span is definitely not beyond conception to think about living a relatively high quality, healthy life, into our hundreds. So,
I don't think we should trivialize that either. I know like there's some pessimism that, just, extending lifespan, seems to be beyond our range right now. I was like, well, hey, I take 120 years in good health, I think that would be a huge accomplishment, So I think, we're probably far away from being able to extend maximal lifespan, but I think we have like so much work to do just to improve, even 10 years or 20 years of improving health span. And I think I'm a lot more optimistic that we have good tools like rapamycin and others that can get us a good chunk of the way there. They just need to be explored further.
I just want to echo that. I think that's such a hugely important point. And sometimes in this longevity community bubble, right, because people have been talking about, immortality or doubling human lifespan, the actual impact of a decade or two of additional high quality life for most people gets underappreciated. And I just want to turn that around and say, just take a minute and think about like, what could you do if you had 10 or 20 years of high quality life? Think about what you were doing 10 years ago, 20 years ago. That's really important at an individual level and at a population level. And we shouldn't lose sight of that, because I agree with Dr. Z. I think that's possible now, or we're really, really close to being able to make that possible.
Sick. Yeah, addressing that
Okay.
health span gap. I think data has been. I want to end it with this one last
One more
question,
question.
just to kind of tie it together, right?
All right.
There was a question about what's in the future for AgelessRx in terms of following up these studies with rapamycin. I know we covered it. We touched upon it within a slide, but maybe what we're most excited about, and then also to Matt in terms of the status of the dog aging project and
Sure.
going on there. I think that's a good question to end on.
Yeah, so for at AgelessRx, I mean, we are prescribing rapamycin for people where it's appropriate to. We do require blood work to monitor patients for safety and things like that, but that also provides us very valuable data. We are planning to start initiatives to look at to enroll volunteers for like metabolomics data, DEXA scans, things like that, to look at our telemedicine users on a longitudinal scale. So, like I mentioned before, we're hoping it's a much larger data set, even though it's not in the in the realm of an RCT. But I think the larger longitudinal data will give us much better insights. And then, like I mentioned, in my talk, we are kind of considering the X Prize and kind of enrolling as a team. I think the current available tools can go a long way in improving function in the 3 domains that they're looking at. Whether or not we have the resources to kind of like to compete, we're still evaluating, but I'd really like to. I mean, I think the work that we've done with the Pearl trial, another thing we didn't talk about was it was also meant to be, our almost like engine of research where, we kind of like start with Pearl, work all the clinical trial bugs out, the software, the partners, the pharmacy partners, all the rest of that. And then once we kind of have that infrastructure in place, then that could potentially open the possibilities for a lot of others. So, I mean, if somebody came to us with like, I think, whatever, like $1 million or a $10 million grant, I think we could quickly spin up the infrastructure to do whatever, pretty much whatever trial somebody would want us to do kind of in the kind of the repurposed drug arena. So I mean, that's the other thing, big accomplishment that I think we came out of with Pearl. And like I said, we have a large database of patient data. We've published one trial about it using low-dose naltrexone quality of life metrics. We have biomarker data and other things like that we'll be digging into. The Pearl just took up so much of our resources. Now that's over, we can then start moving on to like other initiatives as well. So we're just getting started as far as I'm concerned, and we're really optimistic about where we can go over the next few years.
Okay, that's great. So dog aging project, I can give an update. So for as many of you may know, the dog aging project was largely supported by an NIA grant that was not refunded. I won't say anything more about that, except I would use a lot of curse words if I told you what I actually thought about that process. So that put us into a very challenging position. I can tell you today, fortunately, the dog aging project is alive. Daniel Promislo, Kate Creevy, and myself have gone out. We started a 501c3 nonprofit called the Dog Aging Institute. We've raised a little bit more than $1 million to keep the project going. Resubmit 2 NIH grants, one of which for the rapamycin trial, which is part of the dog aging project, got a good score. So I've gone from cautious to cautiously optimistic. I think that trial will finish, which is obviously important. It's been set back a couple of years by this nonsense, but we will get it done. And then we are still waiting to see whether the larger longitudinal study of aging will be funded by NIA. We are continuing to try to raise philanthropic funding through the Dog Aging Institute. If anybody is interested in supporting the project, I would be grateful. You can go to dogaginginstitute.org. We'll be rolling out a campaign next month, actually, which I believe Ageless is actually going to be part of helping us to amplify, and I'm grateful for that as well. So I think it's better, looking better than it was a year ago. I think I'm very hopeful that the Dog Aging Project will be able to continue to carry out this longitudinal study of aging, which has amassed a really, I think, interesting and deep data set that is being made available to the scientific community. And I would also say has engaged more than 50,000 Americans in geroscience research and probably had a bigger impact on the perception of geroscience research within the American public than almost any other project that's out there. So I think there's a lot of reasons why we should all be hopeful that this project continues.
Absolutely.
Okay, well, thank you very much. I mean, again, thank you, Lifespan, for hosting us, and we couldn't have done this trial without the crowdfunding campaign. And thank you, Matt, for kind of supporting us along the way with questions that we had that came along the way. And again, like to thank everybody who contributed, participated, partnered, worked on it. And we look forward to to many more of these webinars that hopefully people will find informative.